You're still here? It's over.
Okay. Hello, hello, hello. I don't know why sometimes when I've not, sometimes it's like freaky stuff starts moving about and changing places, and my mic switched itself off deep within the settings of my streaming service, which is kind of odd. Anyway, yeah, ooh, nice one. Okay, so just going through transport in plants today. So yeah, going through transporting plants. Sorry, I'm sort of fiddling around with my camera angle. It's sitting on the top of my TV right now, but yeah, whatever. You see my dirty washing in the background, or clean washing, but there we go. Ho hum. Right. Let's move on. Let's move on. I should get some sort. I used to have like where I have my background, but sod that. We're not interested in that. We're interested in learning about mass transport. So did a little vote and mass transport came out as the one people wanted to do. I'm doing mass transport in plants today. Fish and insects came a close second. I will do that at some point in the year. I'm covering everything in the year, but it's just like I put it up for the vote sometimes. Okay, so yeah, why do plants need a mass transport system? That is the first thing to learn about. In fact, it's all multicellular organisms, large multicellular organisms need a mass transport system. So this question is a 3-mark question. It says explain why large multicellular plants need a transport system. Of course, multicellular plants couldn't evolve until mitochondria became symbiotic with eukaryotic cells, enabling mass energy production through aerobic respiration, which I will be covering in a future lesson. But that's not the question. The question is, why do they need transport systems? Anyone got an idea why plants need mass transport systems? So why can't that, and what do we mean? What does that mean by a mass transport system? What it simply means is, why can't they let the metabolites like amino acids, glucose, amino acids for making proteins, why can't they just let the amino acids come in through the roots? Why can't they just let the sugars come in, that are made in the leaves? Why can't they let them just diffuse through the plant or move by kind of those kind of mechanisms? So what do we reckon? So hopefully um, you can work out why can't they just let oxygen diffuse through the cell walls and in through the plants to the core of the plant and all over the plant. Obviously, plants are really big. They have a large um, surface area. No, they have a small surface area to volume ratio. So what you want to think about is anything that's large has a small SAV, and this means it cannot meet its metabolic demands by diffusion alone. Okay, it's just too long. It would take 30 minutes for the oxygen to diffuse in, say, and diffuse down to cells for respiration, or for the carbon dioxide to diffuse about to the various cells for photosynthesis, and for the glucose to diffuse to various cells for aerobic respiration. It would take so long to get to certain parts of the plant if it was by diffusion alone that those cells would die. And cells die within a few minutes if they're not supplied with oxygen and glucose. So it's basically, yeah, low surface area to volume ratio is going to be 1 mark. The fact that they're large means they have a low surface area to volume ratio. Diffusion is too slow to meet the metabolic demands of the plants. And if I was answering this, I would always give an example, like I would name a metabolite like glucose or something. Here's the mark scheme answers, so you can check off your thing. Idea of a long distance from external surface to cells, small surface area to volume ratio, diffusion is too slow, named substance. Now, I did mention glucose, and that is essentially what plants want. All organisms want glucose for respiration. Everything alive today that we consider alive uses glucose as the main source of respiration, right? The substrate for respiration. However, you'll hear the talk about sucrose being transported around in plants more than glucose. Of course, sucrose is a disaccharide. It contains a glucose molecule and a fructose, but it's a handy way of transporting glucose around because glucose is more reactive. Sucrose is a non-reducing sugar. Remember, with the Biarritz test, Benedict's test, sorry, you have to add hydrochloric acid, so it's harder to break the bonds and get it to react. So it's a safer transport mechanism for transporting glucose, is to transform it in the form of sucrose. Enough of my whiffle waffle waffle. Nice one, Guy Patton. That answer would get you 3 marks. Okay, so now we've established that plants need a transport system. Let's look at that transport system that plants have. So we're going to have a little check here. You know about the xylem, you know about the phloem. Well, at A level, you have to know not much more than GCSE, just a tiny bit more, but we have to know the names of various bits and bobs in the plant. So yeah, the xylem only transports substances in one direction. The xylem, what are we going to say? Dead cells. Whoops. I don't know if that's an A-level term, dead cells. Essentially, no organelles, no cytoplasm. We want to create no resistance to the flow of water. So the xylem transports water and mineral ions, and we want to create no resistance to the flow of water. So dead cells, that means the only part of the cell they have left, they don't have cytoplasm, they don't have mitochondria, they don't have Golgi apparatus, they have none of that business. The only thing they've got left is the side cell walls, no end walls. So we also, like, they have no end walls, and that helps create a low path of resistance for the water to flow. So I'm not going to type everything out because it's just too long. What's the substance? Now, all plant cell walls have cellulose in them for strength and support. What is the extra strengthening substance that they sieve and companion cells? They're going to be in our phloem. Love it. Does water move via cohesion in the xylem vessel? Absolutement, you're right. And lignin, thank you, Jericho776. So we've got lignin. Now, lignin can come in rings, like you see in a tree. If you chop a tree down, you'll see rings. It's a woody substance, and it's a strong substance. It helps support the plant and hold it up. It does play a role in moving water up as well, but it stops the plant exploding or collapsing, imploding, because you've got this hollow bit in the middle of the plant where the water travels up. Obviously, it's not often hollow. There's normally stuff going up there. So the lignin comes in a spiral form, like a helical, bit like DNA. Like if you imagine a single polynucleotide strand, comes in like a strand like that, or it can just be in straight rings and going around. So essentially, you've got lignin. You don't have lignin in the phloem. Yeah, no lignin in the phloem. Now, depending on lignin, yeah, that's how we spell it, I think. Okay, so I'm going to waffle. I'm not going to write everything down I waffle about, but over here in the flow, what do we call regions? I don't know if you know about these regions. There's little gaps. There's little gaps which are non-lignified. Does anybody know what they are called? Depends on your exam board, really, as to whether you need to know these. But non-lignified regions. There are some regions which are not lignified, tiny little sections that have cellulose but are not lignified. Does anybody know what they are called? And what these things do, these allow water and mineral ions to flow in and out, and they're called pits. Some people call them bordered pits. So these are known as pits, small regions of the wall of the xylem that do not have lignin. They're slightly different than plasmodesmata, which are also holes in the cell wall that allow water in and out. But you've got these pit regions, are specific regions where there is still a cell wall. It's not a hole, still a cell wall, but there's cellulose but no lignin, and they're called pits. Now, if we fling over to the phloem over here, wherever, yeah, probably looking in the wrong place because the way my camera's set up is real dodgy today. I didn't really have time to mess around with it, so it's kind of... It was on an old, where's my clamp and stand? I did have my camera attached to a clamp and stand. Now I've got it on the TV, so you're looking down on my shiny bald head. So you might get blinded if I bend down too long. Right, where are we? Where are we? We've got these regions called sieve plates. Okay, sieve plates are a little bit controversial. All right, they're a little bit of a controversy in transporting plants. The old sieve plates, they're a bit controversial. Now you can see we've got a movement of substances up and down. It's not water and mineral ions, but of course there's water in here, otherwise nothing could, everything has water in it. But this is mainly sucrose and amino acids. So if you're asked to give an example of a metabolite flowing inside the phloem, it's going to be sucrose or amino acids. You've got sieve plates. We've got no lignin in the cell walls. What else? We got companion cells, absolutely. Plasmodesmata, yeah, they are holes, but we've got these special non-lignified regions of the cell wall of xylems called pits. Sucrose moves down, water molecules with it. Mathilde, that is in the phloem, not in the xylem. There's no sucrose in the xylem though. But yeah, so anywho, I'm going to crack on. The main difference is these are still dead. Oh, by the way, I haven't even said what these individual units are called. Very rare that they ask this, but the cells in the xylem so are called vessel elements. Like if you wanted to split it into units where it used to be cells, these are known as vessel elements. What am I banging on about? I'm saying that like each region, you split it into units and they are called vessel elements. Over here, it's a bit easier to see where the units are because you've got sieve plates. So these are known as sieve tube elements. Now the sieve tube is only one part of the phloem. The phloem is made of different types of cells. Someone's already mentioned companion cells. These are called sieve tube elements. Oh my days. Okay, what else did we need to mention? No lignin, movement up and down. Yeah, they have companion cells. There's very little to no cytoplasm in these. And again, not many organelles. They would not be considered, well, they can't live. They can't be described as living cells without their companion cells, which we'll talk about as well. So let's zing on. So now, hey, what exam board? Can you just put in the chat AQA or OCR? Because if you're doing OCR, you've got a couple of extra bits you've got to know on top of AQA. There's just one little bit extra for AQA students that OCR students don't have to know. And there's just a couple of bits that OCRs have to know that AQA don't, if you see what I mean. So it's 80% the same with just a little bit extra for OCR. Because if there's no OCR, I'm allowed the OCR, and yeah. Oh, so we got Jericho's on OCR. Okay, I'll do the bits of the OCR after them. So yeah, everyone's AQA. Most people are AQA. Okay. I'll do the OCR bits after a little bit. So let's check out movement in the xylem and then movement in the phloem. So let's go for xylem first of all. Good question, Mathilde. Are there spaces between the companion and the sieve? No, the companion is touching up to the sieve tube. It's right next door to it. So if I went back up here, I'm going to do a drawing of it all in a second. But if I went back up here, you know, your companion cells would be like that next door. They're not connected, although they are connected. You know, they are touching so that stuff can be pumped and moved through, facilitate diffusion into and out of sort of thing. So they're like right butted up against. There are spaces between cells and there are other types of cells all around. For instance, as again, if you're AQA, you don't really need to know this, but if you're OCR, you gotta know this. There are cells in between. Look, I've got a xylem and a phloem, and there's actually cells in between. Those are some crusty arse cells, but they're known as the cambium, and they're just for support. Cambium, and it's a tissue that is just for support, just for helping keep the plant up, and stuff can move between the phloem and xylem across the cambium tissue. It's also the site of stem cells. So when we need to grow more xylem, these cells can differentiate into xylem or differentiate into phloem sieve tube elements. So the cambium is a supply of stem cells that are mainly for support, but also where differentiation can happen. Now you need to know that if you're OCR, if you're AQA, you can just forget everything I said and just say whatever. Okay, let's zing on and go to how does water move up through the xylem? So check it out, peoples. That is supposed to be a leaf. Okay, so that is supposed to be a lovely green leaf. So if you're thinking, what the hell is that? That is supposed to be a cross section through a leaf. Do you need to know all of this stuff? No, you did it at GCSE. You probably don't need to know all this business anymore. But I've just got it there for explanation purposes. So we have the roots. Now again, if you're OCR, you have to know a little bit about transport across the roots. I will go into that after. But essentially, you've got your root hair cells down here, and we've got osmosis, which is the method by which water molecules enter the roots from a high to low concentration. And of course, they're entering via our lovely root hair cells, you know, the ones that look like somebody with who's got a real long nose. And they have a large surface area for absorbing water, probably plenty of mitochondria for active transport of mineral ions using ATP, all of that business. So we've got root hairs along here, and water enters the roots by osmosis. Now, if you're doing OCR, you need to know about the two pathways that water moves across the roots, because these roots are made of hundreds of well, thousands of cells. So the water has to move across the roots on a journey to get to the stem base where the xylem starts. So the xylem starts at the stem and goes up into the leaf and across the leaf. This is the xylem in the leaf. Okay, so let's look at how the 3 key points you've got to know about how water moves up the xylem. So let's have a little look. So the 3 key points, it doesn't really matter where I start, but I'm going to start here. Water evaporates from the leaf. And we call that transpiration, as you guys know. So water evaporates from the leaf. Oh my gosh, no need for all these notes, but let's put the essentials. So water evaporates from the leaf. Hopefully you know that is called transpiration. Or you can just call it evaporation. And this creates, I could go into how, but it creates a negative pressure. If you want, you can say it creates a pulling force. So your choice of words: pulling force, negative pressure. There's loads of a tension. Oh, let's use the word tension because that's in the theory. So you could say creates tension. Tension, like you're pulling something, okay? Because why? Why does it create tension? Well, if the water's in here, it's all connected in a string. Like the water goes through here and then it's down here and it's connected in a string. So by water evaporating, the kind of string of water moves along to fill the space, creating a pulling force. Okay, so without evaporation of water from the leaves, water couldn't be pulled up the plant. So although we think of transpiration as bad because with transpiration a plant can dehydrate and die, without transpiration we'd have no pulling force to pull water up. So what do we need to know about, I will talk about apoplast and symplast anyway. So this is called the cohesion tension theory. So the first point is explain the tension. So really, if you just have to memorize this on a revision card, transpiration, cohesion tension theory, what it is, 3 bullet points to 4 bullet points, you would say a tension is created by evaporation of water from stomata in the leaf. Boom, right? Number 2, water molecules form a continuous column. So, or let's say a column, a stream if you want to call it, because they call it the transpiration stream. So you can describe it as a stream if you like. A column of water is pulled up due to cohesive hormedes. That's all flicking and flopping about, man. Cohesive properties, e.g. like, so you want to talk about water is polar, bloody, bloody, blah, right? Hydrogen bonding. So I'll probably bust out your H bonding. But, as long as you kind of explain that the water molecules stick to each other and then you basically explain. So they actually move up in like a string. And what we mean by that is you have an O, H, let's just bust that out. Now that is positive. If you're in AQA, you don't need to put the delta symbol, but it's kind of cool. Can't really see it on my crusty drawing there, so I'll do it next to it. But essentially, painful watching me do this. AQA people, you don't need to do that, but you can do that if you want. So what you want to do is express the fact that as these water molecules are pulled up here, each one is stuck to the one behind it, so they're kind of pulling all together in like a prayer set of prayer beads. You see the beads like that, they're all connected, so if you pull on them they're all coming up, okay? And it's because the negative one like that is connected to the positive on the next water molecule. And so what we want to say is the positive hydrogen is hydrogen bonded to the negative oxygen on its neighboring water molecule, and so they are pulled up. And this is described when we're talking about this as cohesion. Okay, so it's why water molecules stick together when they run down the side of a glass or whatever. It's why water sticks together like that. So this would probably get you 3 marks. Water evaporates from the leaf, creating a tension. Water is pulled up in a column because the water molecules hydrogen bond to each other due to the fact that water is polar. And we call this cohesion tension. Now, extra marks, depending on your exam board, but extra marks are for adhesion. So it's not called the cohesion tension adhesion theory, but an extra mark you will often find is for adhesion. And lignin is a charged molecule. It has a slight charge, and so water molecules are also attracted, like Spider-Man when he's sort of climbing up the side of a building. They're also attracted to the lignin in the walls. So adhesion, the polar water molecules can obviously kind of bond to the charged lignin. So adhesion, the polar water molecules stick to lignin in walls, and this is known as adhesion. And that's really it. That's the theory of how water moves up. And if you drill a hole in the stem of a plant and you break the water column, obviously the plant will die because you get air bubbles in there and it messes up with the pressure and it breaks the water column. It's like snipping the string. Any questions about this? Yeah, that hydrogen bonding. Yep, it's a little bit of biochemistry. Too much water near the root hair cells. Well, if you're talking about the root hair cells being flooded with water, then the root hair cells technically cannot get. Someone said, what happens if the root hair cells are flooded? Well, if the soil is flooded, this is a big problem for plants. Most plants can switch to anaerobic respiration, where they might produce ethanol or lactic acid, depending on whether they do lactate fermentation or alcoholic fermentation, to get energy. Because if they can't get ATP, they can't bring minerals in. They could still do osmosis from high to low. That's not a problem. They can still carry out osmosis if it's passive. It doesn't require energy. The problem with flooding is the air pockets in the soil will get filled with water, and oxygen doesn't diffuse very well through water. About, like, I don't know how many 100 times worse at diffusing through water than it is through air. So oxygen diffuses better in air. So you're going to slow the rate of diffusion by about 10,000 times, say, and so they won't get enough oxygen to produce ATP to bring in nitrates and other mineral ions, and then the plant will eventually die. It might be able to last a few days with flooded roots, or a week. I don't know exactly how long. So it all depends. But that's cohesion tension. So let me give you a little question where you would have to apply that and put that in a little question. Let's see if you can do this. All right, let me give you an A-level question. So here's an A-level question about cohesion and tension. So it says, a different student used coloured water to investigate the movement of water in leaf stalks of celery. You might have done this in school. You get some ink in a cup, cut some celery sticks. You can do this at home. Celery you buy for salad and stuff. Stick it in the glass, and then you'll see the ink move up the celery. They cut equal lengths of stalk from each plant, put the cut end of each stalk into coloured water, leave these stalks to take up coloured water for 20 minutes. Use a sharp scalpel to cut slices from the stalks at 1 millimeter intervals until she reached a slice with no coloured water. Figure 2 shows a slice of leaf stalk with coloured water inside groups of xylem vessels. So these are all xylems that are going up like that in tubes, and this is a cross section. Just says, why does the coloured water move up? So for 3 marks, you're explaining cohesion tension. What would be your 3 bullet points? No, I don't do physics at the moment. I'm just focused on the bio and the chem. I have thought about doing the physics, but I think it would be too much on the old plate. So what do you think would be your 3 points as to how water moves up based on what we were waffling about? Cohesion, tension and all of that. And what I'll do is I'll put the exam mark scheme answers up in a sec for that. Yeah, so some people have trouble with cohesion tension, but it really is, what I recommend is putting it on a revision card, it's literally like 3 bullet points. Memorize it off by heart and then spot where you've got to talk about it. So water evaporates, that creates a tension, and then you get the cohesion is the hydrogen bonding. And there's also adhesion to the walls. Yeah, so let's have a little look at the mark scheme, and we'll have a little butchers at this over here. Hopefully you can do this in your head. Water evaporates or is transpired, so through the stomata if you wanted to be funky. This, and you just need to say creates a tension or a pull on the water. Some people, look, it says accept negative pressure for tension. So some people are better with the word negative pressure in their head. It makes it like, oh yeah, I prefer that, negative, like, or whatever. So tension, it basically creates a pulling force. Hydrogen bonds cause cohesion between the water molecules. And if you wanted to go deep, you'd be like, oh, the positive hydrogen on one water molecule has a force of attraction called a hydrogen bond to the negative oxygen on the next water molecule. And again, you should say slightly positive or delta positive and delta negative. And there we go. That would be your cohesion tension theory. So that's kind of easy for the xylem as to what you've got to do. There is also adhesion, but you can see they tend, if it's AQA, they tend to just dash adhesion as, yeah, you can have adhesion if you want. And this maintains a continuous column of water. That's sometimes on the mark scheme that you need to say that there is a continuous column of water. Okay, I'm going to look at the roots and how water moves across the roots in a bit, but I have to check whether you've got to know that for AQA. You definitely have to know it for OCR. So if we have enough OCR people in, I'll zing through the root section. But let me go through the hard bit, okay? So the hard bit isn't movement in the xylem. The hard bit is movement in the phloem. So it's translocation, okay? So this is the bit that people find difficult. And again, I'll go through it all and I'll tell you which bit you have to know for your exam board for translocation. And of course, this is in phloem. It's essentially movement in phloem. And these are all forms of mass transport. So if you get an essay or a long question on forms of mass transport, especially if you're AQA, you get essays, then you've got mass transport of water by cohesion tension, which is a passive process. It didn't require any ATP. And then we've got translocation in the phloem, which is also a mass transport system. And people find this a little bit more difficult generally to understand. So we're going to go through this. So what I'm going to do now is just do an incredible drawing. Maybe not. Oh, it's going to mess with me. It's going to start turning my little wiggly lines. It doesn't like my lines. It thinks they're too dodgy. It starts to make them into things. So what I'm drawing now is a xylem, a very crusty version of the xylem. And you might say, sir, why are you drawing the xylem if you're talking about the phloem? Because mass transport in the phloem involves the xylem. The xylem's only next door. Remember, these guys are in the vascular bundle. They're very close to each other. They're separated by the cambium, which is groups of cells for support that also have meristematic tissue in there, so they can differentiate into new phloem and new xylem. Now, this is supposedly my xylem. Okay, it's a little bit roughly. Okay, so we've got the xylem here. Now, in between, I'm not going to get too creative with the cambium because most of you don't need to know it. So let's just pretend there's a bunch of cells in between. That is some crusty cells. They also give support to the plant. And this is the cambium. So in between the phloem and the xylem. So that's my xylem. This is my cambium. Oh my days. I've got level 3 in GCSE art. Did anyone take GCSE art here? I took art and I made the mistake of thinking that art was like my DOS easy subject. I was like, man, I need to take some like subjects which are just like I don't have to do any work and I'll smash it. What a mistake that was. That turned out to be a proper mistake. Okay, now let's bust out with our whatchamacallit, the sieve tube. So our sieve tube is, check out my sieve tube. What awesome drawing. Yeah, the essays, good practice for the old essays. Yeah, obviously practice is good, but you need to learn the techniques for the essays. Now, of course, what are these dotted lines I'm doing? These are the sieve plates. So if you don't know what they're called sieve plates. and this is obviously my phloem. But is this the phloem? No, this is the sieve tube. Okay, so this is the phloem, but specifically, let's not watch me write out sieve tube. That'd be too painful. This is ST, the sieve tube, and then next door we have the companion cell. Okay, and I'm not going to go too detailed on that. I'm just going to draw it like that. It has a nucleus, it has mitochondria, it has all the things, the rough endoplasmic reticulum, ribosomes, all the things you'd expect to find in a cell. It is the comp cell. I'm going to call it comp cell. It's not a comp, it's companion cell. And we're going to talk about this in a second because we're going to go through the process. I'm going to draw another down here, but does that mean that they're spaced out with 5 in between? No. Each sieve tube element, these are called sieve tube elements, each one of these has its own companion cell, but it would just be long to draw them all out. So I'm going to draw some cells here. Okay. No, Mathilde, on their own, they are not living, but they are more alive than the xylem cells. They have a little bit more cytoplasm and maybe a few ribosomes and a few other little signs of life. But they could not live on their own. They do not have mitochondria, and they don't have enough cytoplasm, so they can't do all the reactions and metabolic reactions to keep themselves alive without the companion cell. So the sieve tube elements are we described the phloem tissue as alive, but if you took an individual sieve tube element, it probably wouldn't survive on its own. But yeah, interesting sort of thing. Right, now I'm going to go through a process called active loading, and I'm going to zoom in. Imagine this companion cell is over here. Let me do it over here because I need to make it big. Over here. Oh my days. Okay, so that is supposed to be a companion cell. Very, very crusty. Now, bear in mind, this is the height of Van Gogh art. This is the cell membrane here, folks. Just to show you, because you might be a little bit confused as to what this dude is drawing. And this is the cell wall. Now, you don't need to know the technical term, but I'm going to give you the technical term. There's a space in between the cell membrane and the cell wall. I don't know if you've heard of it. It's called the apoplast. Okay. There are spaces in the cell wall, so we call that the apoplast. A-P-O-P-L-A-S-T, the apoplast. And so what happens is we have active loading, which is what I'm explaining now, involves 2 channel proteins. So again, if you're doing AQA, this will be very good for you in an essay question, especially as this isn't on your syllabus in this level of detail, so you would smash out the extra marks beyond the spec. So there's a proton pump. Proton meaning it transfers hydrogen ions into the apoplast. Now, you can just say it pumps hydrogen ions outside of the cell. So you can just say that. So hydrogen ions, they get pumped out here. It was actually a dude in the 19th century who came up with this, that discovered the whole membrane hydrogen ion transport mechanisms, which are incredible, is basically vital for all life. So anyhow, this proton pump over here, this is a proton pump, basically a hydrogen ion pump, is made of protein. It's a protein pump. What's it doing? Anything with the name pump in biology does active transport. So that means it's using ATP. Hopefully you all remember what that stands for. It's adenosine triphosphate, and it turns that to ADP. Oh my days. This is painful. I'm watching myself do this and I'm like, it's painful just me doing it, but let alone you guys watching. Right, so ATP breaks down to ADP plus PI. The energy is used to take these hydrogen ions and pump them into the apoplast, the space between the cell wall and the cell membrane. Do you have to specifically say that? You can just say the proton pump, or the carrier protein pumps out hydrogen ions outside of the companion cell. You can just say that. You don't have to say in between the cell wall and cell membrane, but just in case you wanted to use. That's right, Mathilde. They're going to be used with co-transport. So what then happens is a second carrier protein. Oh my God, we've got to draw another one. This is too painful. I'm just going to do a square, right? This is a different type of protein. That is a carrier protein, a protein pump. This is actually a different name. And so again, this comes up when you're talking about transport mechanisms. If you get an essay on transport across the membrane, this is a lovely one for it, because this is called a co-transporter protein. Okay, so this is a co-transporter protein. Oh my days, it's disgusting watching me do this, just doing it myself. Co-transporter protein. Now what happens is, if those of you that have done aerobic respiration, so you've done oxidative phosphorylation, or you've done the light dependent reaction in photosynthesis, you know about chemiosmosis or chemiosmosis, however you pronounce it. That's what happens here, chemiosmosis. So the hydrogen ions are like, boy, we need to get back in. We're in such high concentration. We want to diffuse high to low. And this is a form of facilitated diffusion. It's a type of facilitated diffusion. So you could give this as an example of facilitated diffusion, when hydrogen ions diffuse into companion cells from a high concentration to a low, and an example of active transport, when the hydrogen ions are actively transported out to create a concentration gradient for the facilitated diffusion. So they're pumped out so that they can come back in by diffusion and activate this co-transporter. Now, while they activate this co-transport protein, I don't know how to draw sucrose, because sucrose is like a double disaccharide. That's crap. That's not. Oh, jeez Louise. Right. I can't be bothered to do hexagons. So imagine this is my sucrose molecules. Well, we need to get the sucrose in here, and the sucrose is going to move against its concentration gradient. So imagine this is all sucrose. Right, I'm not going to type. I'm just going to write badly. So imagine this is sucrose. So essentially what's happening, it should be diffusing out, right? Because I've got more sucrose inside than outside. But the co-transport protein, sucrose, is activated and moves in with the hydrogen ions across the co-transport protein. With the hydrogen ions, it's dragged in. Now, this is a form of indirect active transport, because they're going against the concentration gradient, but it does not require ATP. So if it doesn't require ATP, how can we call it active transport? So it gets confusing. So we call it co-transport, because the ATP was used down here to create the concentration gradient, but was used with the hydrogen ions, not with the sucrose. The sucrose comes in because of the hydrogen ions. Now, it's a little bit more technical that in real life, the hydrogen ions actually cause this co-transport protein to spin as they diffuse in. They release kinetic energy, and that is used to bring in the sucrose. So it all gets a bit complex, but essentially this whole process here of getting sucrose into the companion cell against its concentration gradient is known as active loading. So now, who has to know this? If you're doing OCR, you got to know how to explain that. If you're doing AQA, you don't. So you might say, I'm doing AQA, you've just waffled on about that, and I don't need to know this. You only need to know that it's called active loading and that it involves active transport. And that's all you need to know. You don't have to explain that mechanism if you're doing AQA. They can't ask you that in the exam. But it would be useful knowledge for you if you get an essay question that's either on any one of these 5 titles. So if you get an essay question on ions, you're going to get 25 marks in an essay, and 5 of those marks, or a few of those marks potentially, if you've done everything else perfectly, are for going above and beyond the specification. And this would be one example of where you could go above and beyond the specification if you learnt this, and it would apply to maybe 5 different essay titles. There's like a possible 30, and this would cover 5 of them, this little bit of knowledge, because it could cover ions. There's often essays saying describe the importance of ions in biology. If you're AQA, well, you could talk about hydrogen ions here and why they're important, so we can actively load sucrose into the companion cell, and then you could explain the importance that without that we couldn't get this translocation in the phloem, and then plants wouldn't be able to survive, etc. Blah de blah. We wouldn't be able to get glucose quick enough to meet their metabolic demands, all of that business. You could do it with, if it was an essay question on membranes, cell membranes, you could discuss this. If it was an essay question on proteins and the shapes of proteins, you could discuss this. If it was on cell transport, i.e. osmosis active, you get the idea, right? There's quite a few essays that will cover. So anywho, what was this? So active loading. If you're doing AQA, you just need to know that term, and you just need to say, oh, sucrose is actively loaded into the phloem from the companion cell, and that is it. So I'm actually going to go through now the mass transport bit. That's the separate bit of how sucrose gets into the companion cell. So now let's go through. So number one, sucrose, actively loaded into companion cell, then sieve tube. I'm just going to say sieve tube. Oh my days, I need to shrink that down. That's too fat. It's going to be 4 bullet points here. So unlike cohesion, there's a slight bit more to talk about, which is why people find this harder. So number one, sucrose is actively loaded into the sieve tube. So what we're going to do here goes the sucrose into here. And my sucrose should be 2 hexagons, but I'm just drawing it like that. Are there any questions about this? No, this is A level, not triple. Yes, Mathilde, co-transport's also in the ileum, and instead of hydrogen ions, we're using sodium ions in that, and it's the exact same mechanism but with glucose, and there's a little bit more to it. But yeah. Okay, so our sucrose is actively loaded in here. What this does, this lowers the water potential. So by adding soluble substances to pure water, you lower the water potential. So what I'm going to say is number two. So the first thing we want to do is say sucrose is actively loaded into the sieve tube from the companion cell. It's actually actively loaded into the companion cell and then actively loaded into the sieve tube. Now, there are some sources that say it diffuses from the companion cell into the sieve tube, and they'd be correct by facilitated diffusion through a protein. Remember, it cannot diffuse across the membrane. It's not lipid soluble sucrose, so it can't just go across the bilayer like oxygen and carbon dioxide. So it's definitely going by facilitated diffusion. Some sources will say it's loaded again by co-transport, a hydrogen ion co-transport into the sieve tube. So you could kind of say either one really. And then two, the second bit is that this lowers, I'm just going to put initials, I haven't got the kind of space. This lowers the water potential. So water from the xylem moves into, whoopsie, not into the whoopsie, into the sieve tube by osmosis. And I should say down a WP gradient. Now I need to shrink that. Oh my good gosh. Right, that's number two. Remember I said there's going to be 4 bullet points here to explain this process. Anybody got any questions so far about this process? Please do let me know. So it lowers the water potential here, and then water's going to move across the cambium. It's like, oh, hold on a minute. I can go over here because there's a low water potential. So it busses out through the bordered pits or through plasmodesmata. So out the plasmodesmata, across the cambium, by osmosis, down a water potential gradient from high to low into here. Now, what does this do? This actually creates a high HP. Now, HP is that brown sauce people have on fried egg and chips. But in this situation, HP source is hydrostatic pressure. So very similar to the hydrostatic pressure in the mass flow of blood from your left ventricle of your heart. This is the same thing, except for there's no heart involved. Plants don't have a heart, so they have evolved mechanisms without a heart to move substances massive distances. So what we're going to say here is this creates a high hydrostatic pressure. So water and sucrose move down a pressure gradient. Now, oh my days, watching me do all of this. Now you might say, why don't I just have the stuff already up on the slide? But apparently not by my saying, but apparently evidence says it's better to write it all out so people can see how it works. Apparently, I'm not sure about that. It's just long and like longs it all out, to be honest. Okay, so what's going on? Sugar is basically moved in here, lowers the water potential. Water moves by osmosis into the top of the xylem, creating a kind of waterfall, water pouring in. And this is now a physical process rather than, and it's creating a high pressure, and therefore it squirts down the sieve tube towards where there's a low pressure, from high pressure to low hydrostatic pressure. So there's a lot of keywords involved there. So that's probably like a 4 marker to explain movement of substances. Now you could be asked, and I've seen 4 mark questions asked saying, how is it that a low hydrostatic pressure is maintained at the sink, say? So in this situation, this is known as the source. Wherever the sucrose is coming from is the source, and wherever it's going to is the sink. Now, we might say to ourselves, they might ask you, explain how sugars are moved down the phloem. That's cool. You bust out this explanation. But if they say to you, how is a low pressure maintained at the sink, or something like that, then you'd have to explain what's going on here. And what's going on here is that when the sugars get here, they are either going to be actively transported using active loading into cells at the sink, or the sink is respiring, right? So it might be like cells that are respiring, i.e. a new branch in a leaf that's growing, or it might be the roots and they're turning the sucrose into glucose and then into amylopectin and amylose. They're turning it into starch for long-term storage, in which case the concentration of sucrose is decreasing, right? And that means that our water pressure goes up. So at the sink, our water pressure goes back up because the sucrose, the soluble substances, the solutes, are being removed. And what that's going to do is increase the water pressure. And again, what's going to happen? The water then moves by osmosis across the cambium into the, into the xylem, and so therefore maintains a low hydrostatic pressure. If we had loads of water molecules down here, this would become a high hydrostatic pressure and water would stop moving down. Right, any questiones? So Jericho, you nearly got that perfect. What we want to say is sucrose is offloaded at the sink, so the water potential, yes, you've got that perfect, increases. Nice. Yeah, you got that perfect. So water moves by osmosis back into the xylem, and therefore that creates the low hydrostatic pressure. Is the source sucrose from photosynthesis, and the sink? Kind of, yeah, kind of. The sucrose is made in photosynthesis in the leaves, but hold on, it can be either, Mathilde. So let me give you the 2 situations, right? You have to be aware of the 2 situations. So in the summer, the leaves are photosynthesis. This is generalized. Obviously, it can happen at the same time either way. But mainly in the summer, the leaves are producing glucose, not sucrose. They're producing glucose, and then they turn it into sucrose for transport, right? And where are they sending it? Well, they know they've got a long autumn and winter ahead where they won't have any leaves. Now the trees are losing all their leaves. And so they want to send the sucrose to the roots, which would be our sink, where it's going to be turned into starch. Okay, and of course starch is not soluble, so it's not going to affect the water potential. So that's why the water potential is increasing down there when you're turning the sucrose into starch. So, however, in the winter, it could be going anywhere, right? In the winter, our source could now be the roots where the starch is, and they break that down into glucose and then turn it into sucrose for transport. And the sucrose then moves up the phloem and maybe goes to a branch. I don't know why it needs a branch there in the winter, but whatever, to keep that branch alive. So it needs to respire, right? So the source and sink can flip round and be in either direction. But yes, the source is wherever sucrose is being made, and the sink is wherever sucrose is being used. And it could be getting used for respiration, or it could be getting used for storage, or to make berries like fruits or strawberries or something. So anything like that, yeah. Now, anybody got any other questions about that? That's called mass flow. Now, you got to know about evidence for this. I might just waffle, but just make sure you're aware. I would have a revision card and have evidence for mass flow. So the evidence for mass flow, if you're doing AQA, you specifically have to know a couple of pieces of evidence for mass flow. And so the evidence for mass flow is, number one, something called respiratory inhibitors. These are enzyme blockers, right? Competitive or non-competitive enzyme inhibitors that block enzymes involved in the Krebs cycle, right? And you've heard of cyanide, you've seen it in the movies, Captain America or whatever, or Marvel. People take a cyanide and they die. Well, what does cyanide, the poison, do? It blocks it inhibits some of the enzymes involved in respiration. So if you block respiration in the companion cells or in the phloem, what happens is without ATP, this doesn't happen. So without ATP, transport along the phloem doesn't happen. Now, how do we know that transport doesn't happen? Well, because we can radioactively label sucrose with carbon 14 or carbon 13. We can use a radioactive bit of carbon and we can trace the path that this radioactive carbon has gone. So we can prove, therefore, no one's actually seen that. This is still a little bit mysterious, and there are arguments about this. Okay, so it's not 100% solid theory, this active loading mass flow business. It's not 100%, it's mostly 99% accepted. It's not 100% accepted but we have evidence. So you have to look at the evidence for and against. So the evidence for is that if you stop ATP, you stop mass flow of sucrose in the phloem. So that means we know it involves active transport, and so someone come up with this mechanism based on that. Now, some of the other evidence that sugars generally move from the source to the sink like this, and due to high pressure. So, and then you've got the whole, well, how do we know they drew to pressure? It moves due to a high pressure to a low pressure. There's one where they take green fly aphids and they use their little mouth, proboscis or whatever it is, to puncture the top of the phloem and the bottom of a stem, and they find that the sugary sucrose liquid spurts out at the top and it trickles out at the bottom, and that tells them that the pressure is higher at the top. So if you put a hole in a stem at the top and a hole in the bottom using an aphid, now you can't just use a pen or pencil to do that and prove anything. Aphids have a perfectly evolved proboscis for piercing the phloem and the depth of where the phloem is. And so what they find is that it comes out under pressure higher up, which tells us there is higher pressure at the top. So again, that's more evidence. And then there's some radioactive evidence. You can kind of get a plant and you can use radioactive isotopes again inside the sucrose, and you can use X-ray film, and as the sucrose goes past, it decolor up, makes the film go black essentially. I don't know if you get a 3 marker on the tracing. I'll have to see if there's any. I haven't seen any questions on it, but it's definitely on the spec. Yeah, also ringing. Is ringing where you cut a bit of it and it bulges at the top? What's ringing, Oscar? Is that where you get a bulge above the ring? So you remove the bit, or you take a tree and you remove the piece with the phloem in, and if you do that, it bulges above the ring. That might be what you're talking about. And that's where, again, proving that there's you develop a high it moves due to a pressure system. Yeah. So yeah. And so a bit of AQA, a bit of OCR. Now again, if you're AQA, remember you only have to know the active load. You just have to know the term active loading, and you just have to say sucrose is actively loaded into the sieve tube. Now most of the time in OCR, they will ask the questions about this separately, okay, to this. So they'll have like a 3 marker or 4 marker about how does mass flow or mass transport of sucrose from the source to the sink work, and you just reel off this bit here. Or they will specifically have a question saying explain how sucrose is loaded into a companion cell, and then that's where you'll bust out this and you'll say hydrogen ions are moved by a protein called a proton pump into the outside of the companion cell. You don't need to say into the space between the membrane and the cell wall called the apoplast. You don't need to really say that. You can just say hydrogen ions are pumped against the concentration gradient using ATP would definitely be a mark to ADP, and then you'd say they move back in down a concentration gradient by facilitated diffusion, or you could just say they move by co-transport through a co-transport protein bringing sucrose with them. The sucrose moves against its concentration gradient. And you just mentioned the co-transport protein. And this process, if you wanted, on the mark scheme would be chemiosmosis. It's the process by which hydrogen ions are actively pumped out and then move back in down a concentration gradient. Okay. Let's have a little look. What else? Have we got any OCRs? I haven't really done the specialist bits for OCR. Let me see. What's this question? Oh, there we go. So here's a 4 marker. See how many marks you can get on this 4 marker. It's just 4 bullet points. Describe the mass flow hypothesis for the mechanism of translocation in plants. You do not need to talk about the active loading. They will make it specific when they want you to talk about that. So see if you could write down in the chat, just put your 4 bullet points, just 4 bullet points. Remember, in biology, you can just bullet point your answers. All right, as long as they're... and if you're on the AQA, you can literally put bullet points. They don't even mind if you actually do little black circles. But you want to put your answers like that. I mean, yeah.
Stick around.
I'm gonna have to science the shit out of this.
Okie dokie. This is relevant to all exam boards, by the way. Where are we? Hey. Okay, I'm going to put the answers up in a second so that I can move on. And I'm going to go over just a couple of bits. I'm going to see, is there any OCR people left in the chat? Because I'll bother doing the... yeah, so Matilda's got an answer. What's this? Active loading of sucrose into the sieve elements from companion cells. Yeah, smash. That's going to get you a mark. And then... Okay, Oscar, sucrose actively loaded at the source by companion cells, lowers the water potential at the source, water moves into the phloem from the xylem via osmosis. Yep, this creates a hydrostatic pressure gradient. Nice. I don't know whether you'd have to have said water moves by osmosis down a concentration gradient, but it's kind of implied in the name osmosis, right? So, and sucrose moves to the sink where it is actively unloaded. Yep. Yeah. Here's the answers, guys. I'm going to put the answers up for you guys. Nice. Like it. So, in the source, leaf sugars actively transported into the phloem by companion cells. It might even be 2 marks, or maybe that's 1 mark. No, I think that's 2 marks. So that's a bit generous, isn't it? Actively loaded into the phloem by companion cells. They haven't even said sieve tube. Okay. Lowers the water potential of the sieve tube. Water enters by osmosis. They put, see, isn't that weird? They put lowers the water potential of the sieve tube and water enters by osmosis from the xylem as the same marking point, and yet they put active loading from the phloem by companion cells as 2 separate points. So each one of these lines is a separate point. Okay, fair enough. Increase in pressure causes, yeah, so explain that we've got an increased hydrostatic pressure, or just an increased pressure, causes water to move down a pressure gradient towards the sink. And then they've just said sugars are being used in the root for respiration for storage. I mean, that's not necessarily fully accepted. Oh, they're saying except that it's converted into starch for storage. But it might be that the root is respiring. So, and it didn't specifically say to the root. It's just the annoying thing about exam questions. It's very vague, because it could be that maybe I've only cut part of the question out, and in an earlier, I don't know, 7II or something, it was talking about the roots. But there we go. Your answers sound pretty good. And we'll get you all of the marks. Right, let's just check out what is this one. Okay, so here we have a dirty, nasty question. Oh, it's an evaluation. I don't want to do this. Never mind. Okay. Block of egg. Oh, let's not look at this. I'm not in the mood for that right now. I'd have to actually think about this, and it'd be painful. what I wanted to say is that is there any OCR people, because I didn't go through the Simplast and Apoplast pathway. I don't think I've ever seen a question on that in AQA, so I don't believe you need to do the Apoplast or Simplast pathway in AQA. But yeah, there is something called the Simplast and Apoplast pathway, and it is the way that water moves across the root cortex, so the cells of the root, and across the cells of the leaf to get to the spongy mesophyll from the vascular bundle, and then turn into water and evaporate. So, but yeah, if there's someone from OCR, I'll bust that out. I'll bust through that. But this is a nasty question. I haven't even thought about. Right, well, we've got a block of agar there. Right, so they've cut the top off the sieve tube. Right, block of agar, no substance X in the agar. I don't even know what substance X is. Section of the stem is intact. Substance X in the agar. Middle of the section of the stem intact. Oh, I think I'm going to allow this. I'll do this one later. Potentially. I think I did that last year. That's one I went through last year. All right, let me go through. This is strictly for AQA and OCR. If you do AQA and OCR, then you need to know about this one, which is dissecting plants. So both of you need to know this. Very, very easy. What's this lady doing? Is she using a scalpel? Use a sharp scalpel to cut a cross section of the stem. Dip them in water using tweezers so they don't dry out while you're preparing all your bits, getting your cutting board ready, and you're doing your whatever you're doing. So it says, like, if you're going to do a dissection, put the pieces into distilled water so they don't dry out. Okay, and then you're going to stain it with TBO, capital TBO, which is known as toluidine blue O, and leave for a minute. The TBO is charged, so it will stick to the lignin, and they go blue-green in colour, and that's all you got to know. So you're likely it'll be a one marker, and you just need to say use a scalpel, cut it open, dip it in TBO. Okay, you need to know that for OCR and for AQA. Now, if you're interested in how water moves across the roots, I'm going to quickly let me just bust through how water moves across the roots. So if you're interested in how it moves across the roots, oh my days, I'm just going to quickly bust through that. So just stole this image for educational purposes. I didn't steal, I borrowed this image for educational purposes. I could have drawn it, but you all saw how long it took me to draw out the xylem, phloem, cambium, all that, so we didn't want to wait for me to draw this. Right, so here you've got your root hair cell. Water moves in by osmosis. Now, there's actually three methods by which the water crosses the cells of the roots. So if you imagine a root, we call the cells in the root cortex. Okay, so it's going to move across those cells in the root. The final layer of cells in the root cortex is known as the endodermis layer. It's going to move across in three ways. They've only got two ways here, but there's another way called the vacuolar pathway. You don't need to know too much about that, other than that water travels between the vacuoles of the plant cells. So there is a pathway where water travels from cell to cell via the vacuole. That's the vacuolar pathway. The two main ways you have to know: there's an apoplastic pathway. Do you remember I mentioned earlier that apoplast is the spaces between the cell wall and the cell membrane within a plant cell? There are empty air spaces. Okay, and what that means is the water gets sucked along via a pressure gradient. Okay, it's kind of diffusing from high to low, and it's moving along the cell wall, and it does that through the root cortex. We call that the apoplast pathway. Then we have this red pathway. I hope you can see that on the screen. We've got this red pathway. This is known as the symplastic pathway. Now, this big fat white circle in the middle of the cell is the vacuole. It is not the cytoplasm. The light green material here is the cytoplasm. So I think phonetically the symplastic pathway, which is spelled SYMPLAST, symplastic, sounds like cytoplasm. So that's how I remember symplastic, cytoplasm. So the symplastic pathway goes through the cytoplasm. Now, how does it cross from one cell to another? Well, it moves from a high water potential to a low water potential by osmosis. So the symplastic pathway is moving by osmosis from cell to cell down a water potential gradient, and how does it get through the cell wall? There are small small holes in the cell wall that join the cytoplasm together of various cells, and it's called the plasmodesmata. Okay, now, yeah, sorry, I was taking a breath because I was talking a bit too much. Right, so get that down. If you don't know that on a revision card, you can put that apoplastic pathway is through the spaces within the cell wall. It's via diffusion and pressure. And if we look at the symplastic pathway, phonetically sounds like cytoplasm. It travels through the cytoplasm by osmosis, cell to cell, down a water potential gradient via the plasmodesmata, which link the cell walls together. They're tiny holes that link the cell walls. Now, when they get to the endodermis layer, if you are in OCR, if you get to the endodermis layer, the apoplastic pathway hits an area called the Casparian strip. It is like, whether it's made of like a waxy lipid, it is made of an insoluble substance. And therefore, the apoplastic pathway ends at the endodermis layer. And the apoplastic pathway is forced in to the cytoplasm, where it joins the symplast pathway and they become one. So it's like right at the base of the stem, the two pathways crash together to make a giant waterfall. I guess this increases the water pressure, and we get a little bit of upward pressure to get the water moving up the xylem in cohesion tension. We get a little bit of a splish splash as they come together, okay? And that's the endodermis layer. It contains Casparian strip. The Casparian strip only runs through the cell walls. And what else do we need to say? These are, you might have heard of these. These are parenchyma cells. You might be like, what? Have you heard of parenchyma? Parenchyma cells are just any normal plant cell. All right, it's just any plant cell. Palisade mesophyll, spongy mesophyll. You remember them from GCSE in the leaf, the spongy mesophyll, the palisade mesophyll. Those are just known as parenchyma cells. They're just your general normal adult plant cell. It's just given a funky name. Do we need to know about the Casparian strip for AQA? I don't believe you need to know about the Casparian strip for AQA. No, I haven't seen it in the CGP. I probably need to check the spec. And I haven't seen it come up in any exam questions for AQA, the Casparian strip. That's not to say that it wouldn't come up in the stem of a question, but where you wouldn't be expected to know it off by heart. You might have to apply your knowledge and say, oh, there's this path. I have seen a question in AQA where they talk about the apoplastic pathway or the symplastic. Only one or two questions, but they were not expecting you to know anything about them. They were expecting you to give you something in the stem and then you apply your knowledge to it. So it wasn't something you had to know. You might hear as well, if you're doing EdXL or OCR, you'll hear maybe of sclerenchyma cells. So they are cells that are in the stem of the plant and they are just for structural support only. It's a tissue that just has one purpose, and that's to hold the plant up. They're known as sclerenchyma cells. Would these pathways also happen in the leaves? Absolutement. So if we go to the leaf, let me go up to the leaf and show you. Where's my leaf? It's over here. What we want to do is steal this little image over here and let's zing this out. Okay, sound effects. So what's going to happen here? This is the vascular bundle, and out comes our water, right? Yeah, out comes our water, and so how's it going to travel around the leaf? I haven't really got good cells here to travel around, but essentially it's going to apoplast like that through the cell walls on its way to the palisade so it can do photosynthesis, right? They want to get in the chloroplast. At the end of the day, the final destination of H2O is supposed to be the chloroplast, right? It's needed for hydrolysis. What's it called? Photolysis. Photolysis to supply electrons to the electron transport train and hydrogen ions for the dreaded chemiosmosis in the light-dependent reaction. So, and for reducing NADP to NADPH. So that's what water's needed for. But it's also going to go through by the symplast pathway like that as well. So when it gets through cohesion tension up the xylem, it will return in the leaf to traveling across these cells like that. So your answer is yes, they will travel by those pathways across the leaf. Remember, they can't travel in those pathways up the stem because the stem is dead cells and is a physical process. Now this tells us plants had to have mass transport to evolve, right? Because they couldn't, what it's telling us is, look, in order to get the water from the bottom of the stem to the top of the stem, it couldn't rely on the apoplast and symplast pathway. They just aren't quick enough. The plant would be dead. It wouldn't get its metabolites, its minerals, its magnesium and whatever else it needs fast enough relying on that. So it needs these mass transport, and all mass transport methods involve pressure gradients. They involve physics, physical processes like with forces and pressure, because you can't rely on these kind of biochemical pathways, diffusion and all of that, or osmosis. Osmosis and diffusion just ain't gonna cut it. So it relies on a bit of physics to keep the plant alive. Okay. Anybody got any other questions about transport in plants? Oh, if you are OCR, you might see some of these lovely drawings. So if you're doing OCR, you might see these drawings. Sclerenchyma are just structural, and then this red bit, oh, you can't really see that. My face is, my body is over the red squiggle, but the red bits are the xylem. So that is the cross section cut through of a stem. The red bits are the xylem. Excusez-moi. So the red bits are over here, the xylem, and the blue bits are the phloem. And then over there, oh, this must be a root. Sorry, this is a root tissue. So, oh yeah, there's root hair cells. Kind of gives you a clue, right? There's root hair cells over here. So this is the xylem, and then the phloem is these little patches around here, and then you've got kind of sclerenchyma all around, which is just structural support, and parenchyma, which is just normal cells. But yeah, if you're doing OCR, you need to know all these kind of these disgusting cross-section diagrams off by heart. So yeah, that's kind of it really. Let me just see. What are these pathways happening? Anybody else? So the phloem is on the outside of the stem. That's why they do those ringing. Yeah, that's absolutely right. Yeah, the phloem's on the outside with the stem, but when you bust into the root, yeah. The only bit where the phloem and xylem get a bit confusing is, I always think, is the cross section of the leaf, where the xylem is on top and the phloem underneath. Doesn't make sense compared to the rest of it. So in the rest of it, the xylem's on the inside, whether it's root and stem. Xylem's kind of inside, phloem outside. I have seen questions, if you're OCR, I have seen questions where they zing on that. And they say, like, what is the tissue in between the phloem and xylem? And of course, that is, oh my days. Check out the writing. That is cambium. Is that an M? I don't know what it is. Yeah. Oh, like, so you got like this diagram showing the vascular bundles in the transverse section of the stem. So this is the stem. And like you said, they do the ringing experiments. So because you said the ringing experiments where they cut the outside of the tree, that means that R must be the phloem, right? Tell me R's the phloem, right? And so the xylem must be U. It's on the inside unless it's the leaf, I always think. T, God knows what T is. The cambium would be, V, I believe. I hope. Hopefully we got that right. U-R-V. Cambium should be the cells in between the phloem and xylem, because it's the kind of bit that they flow across during osmosis. So, yeah. Oh, look, there's the answers. Yay. Any questions, peeps? Otherwise, I'm going to finish there. Now, does anybody have any questions about anything they're doing in biology at the moment, just for the last couple of minutes? Anybody got any questions about anything they're doing in bio, or anything they'd like me to go over next week? I actually have loads of questions on here, but I'm a bit knackered today. So, anyone got any questions? Cool. All right, guys, I'm going to shoot off. So, yeah. I've done a walkthrough of the AQA Paper 1 A-Level Bio 2024. I'm going to do a walkthrough of the Paper 2, hopefully before Christmas. Probably not in time for people's mocks, but you probably already had your mocks, right? Has everyone had their mocks for A2? But I'm going to do like, I've still got to do a walkthrough for the A-Level Biology Paper 2 and 3 for 2024. And I don't know whether to do the OCR ones. I don't know if there's enough people doing OCR. Like, it seems like most people seem to always be doing AQA, so I always like prioritize AQA. But yeah, Anywho, all right, I'm going to shoot off. Yeah, I need to get the chem ones done. I'm working on the paper 1 chemistry, which should be out next week if you're an AQA chemist. And then I might do the OCR one, or I might do paper 2 chemistry the week after that. So Mox in Jan, cool. All right. Yeah. Well, I should have. Are you doing chemistry as well? Do you do chemistry and bio, or just bio? All right, nice one, guys. Yeah, let me shoot off. Okay. I don't know what's going on with my thing today. All right, take care. Enjoy your evening, guys. Keep working hard. A levels is all about consistent hard work. Got to memorize so much content. You just got to revision card it, memorize it. You got to be working now. If you want to get an A or an A star, you've got to be really working now. Like you haven't got time to lose. So if you haven't been at full speed, you need to be revving up to full speed, and you need to be like entering like full working mode. You've only got 6 months, kind of. So nice. All right, guys, have a good one, yeah, and keep up the hard work, and I will see you next week.
You're still here? It's over.